topics that you've been hearing earlier in this conference and concentrate a little bit on how are we going to make money by going to space. And at this point in time, I think we recognize that we can make a lot of money if we're in the communication business, and we'll probably make a little money if we were in the remote sensing business. But I do think that as we move into the 21st century, we're going to see some things happening that are going to impact our quality of life and our quality of environment by using plants and the research in a little different way than what you heard earlier. There's no question but what plants are a major part of a life support system. On Earth here, they function in that category as well. But in addition to that, I think that they are our source of food and fiber, and we'll probably look at many different kinds of uses for plants. So as we look into the next century, I think we are going to see a major impact on quality of life here on Earth as a result of having done a couple of things in space. One of those being the ability to insert unique gene characteristics into plant material that make it much more of an economic value. I think we're going to be growing crops for other purposes than just food and fiber. I'll just touch on that from the standpoint that you may or may not have seen reference in one of the national news broadcasts a while ago where the banana plant had been, the gene had been inserted for resistance to diseases. So instead of getting a flu shot, you would eat a banana and have the same effect. So that's only one part of it. The industrial uses that we have potentially are also there. And then finally, I think the other thing that we're going to look at in the 21st century is increased use of controlled environment agriculture. That doesn't mean that we're going to replace the millions of acres of corn or the millions of acres of wheat or the millions of acres of cotton. But we're going to do specific things which are going to enhance our ability to use those millions of acres so that we can produce more food and better fiber, and other uses. To give you an idea of the impact of genetic engineering on the agriculture of this year, and this is a chart that appeared in the Wall Street Journal about a month ago. But I think the almost unimaginable thing is that the growth in the acreage of cotton that will be planted to genetically engineered varieties is essentially 50% anticipated this year. Now, half of the cotton acreage will be planted to genetically engineered materials. And that increase has occurred over the last 3 years. The same thing, you can see the trends in soybeans and you can see the trends in corn. The other significant thing about that chart is that that acreage is due to the insertion of primarily two genes. One is the gene for Bt resistance, or the Bt gene, which imparts disease resistance to cotton, disease resistance to soybeans, disease resistance to corn. The other is a gene that allows the farmer to spray a herbicide that controls a vast array of plant materials, but not that particular crop. And so those two genes, and if we look at that biological diversity that exists in really the plant materials we have and the potential for this kind of thing, when we can see that the impact of just two genes, what will that mean in the future? And so we are already experiencing a considerable amount of this kind of activity. And how does space enter into the picture? Microgravity, we know, affects plant materials, biological materials, probably animal physiology much more than plant physiology, but because of the fact that we do not have the thermally induced convection of cell materials. In other words, where you have the heating and cooling of cellular materials or the heating and cooling of a liquid, as you know, boiling water. The other thing is that the cell component does not sediment. There are little bodies in the cell that will sediment out here on Earth unless we physically disturb them. That sort of sedimentation does not occur. Mass transport within and between cells is definitely affected. And finally, the allocation of energy among secondary metabolites. How does that kind of thing affect or impact our ability to insert a very foreign gene into an economically important plant material? And so we're looking really for two things in that process. One is to increase the efficiency with which we can do that, and secondly, to regenerate some of the tissue that we need to have as we do that process. Now, I'll throw this up, and if there are any molecular geneticists in the crowd, I would apologize for it, but I think it illustrates the sort of thing that I'm just talking about. This is one way that you can take a gene from a very distant material and put it into a corn material or a cotton material, or for that matter, a flower material. Basically what you do is you take a leaf disc, sterilize it, and then finally expose that leaf disc to a bacteria that carries the gene, or the vector that carries the gene that you want to put into this tissue, leaf tissue, which happens to come from corn plant, cotton plant, whatever. And you allow that these leaf discs to be infected. And as that leaf disc repairs the cells that you hurt or damaged by cutting it, some of those new cells will incorporate the DNA from the bacteria that carry the desirable gene. Some of them. And what I'm saying is that we anticipate that if we do that process in microgravity, more cells will have that new gene incorporated in that particular process. And there's another step. So now you've got a leaf disc, and if you culture that, you'll get a clump of cells. And if you culture it in the right way, and by using an appropriate material, you will kill those cells that have not incorporated the new gene and leave the cells that have incorporated the new gene alive, so that you have a callus now that has the desirable gene. A callus of corn tissue, a callus of cotton tissue, or something like this. What you need to do is then regenerate that into a plant that has leaves, stems, roots that you can use in a new varietal development program. That regeneration is a very critical step, and is a very difficult step, impossible for some of the species. We feel that in microgravity, this regenerative process, moving here, would also be enhanced in microgravity. Turning to the other part of another area that we feel is very important, and that is the development of new plant materials. Fundamentally, what it amounts to is that we'd like to understand how environment affects the biosynthesis of critically important and economically or commercially important parts that we really want out of that plant. And we heard the various things about what kind of things are we looking for. We're looking for the production of an alkaloid that is a medicinal property. We're looking for the production of an enzyme that we can use in a industrial process. And so if we understand that biosynthesis, and I'm sure that we study these kinds of processes, how environment affects us, microgravity is still one other environmental factor that will help us understand that. The other thing is, very likely that we look at this plant material carefully, we may find that this biosynthesis in microgravity will give us a compound that is different than that we find here on Earth. And again, we have a history of understanding that a minor change in the biochemistry of that particular material has a great impact on its ability to do what we want it to do, whether it is a drug or some other kind of material. And finally, I think we want to know how environment and other factors affect the storage of this. In other words, how much material can we get so that we can increase the percentage of the vital, commercially important part that we want of that plant instead of producing a lot of other things. And finally, I don't know why, it looks, I think it's because of the parallax, but it looks real square here. Until I look at it, it doesn't look square. And finally, I think what we're looking at is the facilities that we need to grow plants in space, more particularly in low Earth orbit, where we have a very low level of gravity. We need to have appropriate plant growing facilities. And the program that I've been involved in, together with our industry partners, we have developed some of the facilities for growing plants in space. And that is that we have to have the ability to control temperature and light, humidity, and CO2, so that we are looking only at microgravity, or if we want to, the interaction of temperature with microgravity or humidity with microgravity. And to date, most of the hardware that we have used and have that available to us is not adequate to really separate that out. The other thing is that, and so I mentioned the use of genetic engineering where you're inserting a desirable gene into a material. You really have to stabilize that in a commercially important material or a variety. And usually the seed or the seed production, people like to make a number of back crosses to stabilize it into the agronomically important or agronomically useful material. And that takes a number of generations. We can do that in the controlled environment facilities that we're developing for use in space. And finally, also the kind of technologies that we are using for growing plants in space can be adapted for the production of high quality plant material. And I'll just go through some of the details of a unit that we have been using for growing plants in space. This is a unit that we've flown on the shuttle. It's a single mid-deck rocker, and we have an exhibit out here. You can see that particular unit that was up there on SDS 73 in October or November of '95. It has all of the controls for maintaining temperature, humidity, CO2, provide the light that we need to grow the plants, and then provide the water and the nutrients to the plants. The experiment that we conducted on that during that mission was a 16-day mission in which we took a cutting or a leaf of a potato plant, and if you treat it properly, what you'll really result is a little tuber that you can see at the base of this leaf after a period of about 10 or 11 days. That particular slide illustrates some very fundamental things that have not been really clearly demonstrated as the impact of microgravity. Number one, that little leaf was able to conduct photosynthesis. Number two, that leaf was able to translocate that energy from a leaf to a bud. That particular bud was able to develop into a storage organ, and that energy was stored. So that most of the functions that we look at here in the on Earth, we are able to say actually happen up there on in microgravity. That little unit does not allow us to do very much plant work. So we're looking at developing a unit for the International Space Station, which is basically installed in one half of a rack. And the growing chamber that we have in the unit that we fly on the shuttle is basically a 5-inch square by a 9-inch height. That's our growth chamber, or that's our greenhouse on the shuttle. This one we were looking at will give us an area that's much larger, which is 20 by 20. We can keep it up there for longer periods of time and give us the kind of insight into the things that I was talking about. I just wanted to show you this particular picture of how those facilities can be used here terrestrially as well as in space. This is a crop of soybeans that was grown from seed to maturity basically in 9 weeks, or 63 or 64 days. That same crop in the field would be take somewhere between 100 to 110 days. This means that the seed company interested in developing a new variety of soybeans could actually go through 5 generations a year. Currently, they go through 1 generation here in the Midwest, take the material to Puerto Rico, take it to Hawaii, take it to Mexico, take it to South America to try to get 2 generations a year. You can see that the advantage in really meeting the marketplace is significantly enhanced by the use of the kind of controlled environment facilities that we've developed and needed to develop for growing plants in space. This is another variation of the same sort of thing. This is a potato seed production system. Potatoes, unlike corn, are propagated by the tuber. It's a little vegetative cutting. And if you've got a virus in the plant, it's in that tuber as well. And so over a period of time, the seed that you use is inferior and will not give you the kind of production on a commercial field that you want to. When I was a kid growing up on a farm, we would set aside a bin each year as our seed stock. If you did that today, you would probably last 1 or 2 years. Yes, thank you. Why? Because your cost of production is such that your level of production must be at a high level, and so you go back to very high quality seed. In this case, what we're doing here is working with a company that is using the controlled environment technology that we developed for growing plants in space to grow small cuttings of potatoes that produce a mini tuber. That little mini tuber can then be moved to the field, and the next year will be the production of the seed that will be planted in the farmer's field, so that that farmer really comes back to very highly elite seed materials, that has been exposed to the ravages of the field for only 1 or 2 generations at the most. And finally, I'd like to just touch on the experiments that we are planning to conduct on a shuttle mission that will be flying in this late October this year. We will do a gene transfer experiment on some soybean materials to see whether we can enhance the genetic engineering process by doing this in low Earth orbit, or where there is a reduction in gravity. And then we are also going to be conducting an experiment to study the biosynthesis of a secondary plant material, which happens to be an volatile essential oil. It's a model. And if we can show that the biosynthesis is different, it leads us to a number of things that we can do to really make money by going to space beyond just being a communications organization or a remote sensing organization. Thank you very much. Yes.
Right. I read somewhere that lignin production of plant in space is reduced, and therefore more of the material to go for the food going here in the part of the plant. Is
that's the evidence that is in the literature at this point, which means that instead of producing lignin, that energy is being used for some other things. And that's what comes back to understanding the biosynthetic pathways and how biosynthesis is affected by microgravity. That's a classic example.
Years ago, I attended a lecture, I think by a Swiss expert. He referred a great deal to a Russian expert. Their mission was to look for original seeds around the world. Now, if I remember correctly, there was some concern about moving to these various generations of hybrids and so forth. We had to want to keep an original seed stock somewhere.
Yeah.
What is the significance of that?
Okay. What that means is that if you go back where wheat really was originally grown, where corn came from Central America, you'll find probably the greatest diversity in corn. And so you can go into corn and try to pull that out into the corn that we grow here in the Midwest. In the case of the BT gene, that is a bacteria. And so we know that bacteria, when it infects an insect, it kills that insect. And so what has happened is the genetic engineers have taken that gene out of that bacteria and put it into corn. So corn produces the same protein that would be produced by this bacteria. So it's a little different kind of thing. You find a gene way out here, whereas the kind of thing you're talking about is extremely important. You go back to where the species originated because that's where the most diversity occurs. Yeah.
At this point, you're looking at just active, interested in producing
We're interested in producing a large amount of active. The research will be done here on Earth. And so when a seed company says, I want to transfer this gene to the existing soybean variety, we'll do that up there, bring it back here, and then put it into their.
How is this coordinated with a lot of
I'm sorry?
How is this coordinated commercially with a lot of consolidation?
Yeah, what will happen is that basically, and I'll use the name, Pioneer Seed Company will pay someone X dollars to do that for them. And that will be done on the station. That is what I consider to be truly commercial use of space.
How successful have the Russians been using the rear to do plant transplantation?
The reason why I'm struggling a little bit is limited. Limited because the unit that they have up on for doing plant research on the Earth is quite inadequate. It has no temperature control, it has no humidity control, it has no CO2 control, it has very polite levels. And all of that, when you put that into the mix, the results you get out are very difficult to interpret.
Is this material you give, is that a preprint or something like?
There is,
Summary of that?
yeah, there was material. I did submit a paper to the people, so it should be in there. If not, I think I'd be glad to provide it to you.